So we just discussed how water is able to freely move between body compartments, but what about other particles? Humans are very large, complex organisms, and the movement of materials, nutrients, or other particles within and between body compartments is necessary for communication. And this movement requires a variety of transport mechanisms. So this section of the lecture is going to describe and discuss these different transport mechanisms across cellular membrane in between different body fluid compartments. So this biological transport is another theme that you will encounter repeatedly throughout the semester as we study the different organ systems. The most simplistic and general form of biological transport is called bulk flow. Bulk flow of fluids within a compartment is where the pressure gradient causes the fluid to flow from regions of higher pressure to regions of lower pressure. And as that fluid flows, it carries with it all the subcomponents, and including the substances that are dissolved or suspended in it. For example, blood moving through the circulatory system, right? We've got blood proteins suspended in it, blood cells suspended in it. So during the bulk flow of blood, all that is moving along with that plasma. Airflow in lungs is another example of bulk flow. And that's something we'll discuss more when we study the respiratory system. Other forms of transport are more specific and a little bit more complex than bulk flow. And when we discuss them, we have to name the molecule or molecules that are moving. And transport mechanisms we'll learn about include diffusion, protein-mediated transport, and vesicular transport. While many materials can move freely within a body compartment, exchange between the ECF and ICF, that means extracellular fluid compartment, and the intracellular fluid compartment, exchange between the two is restricted by the cell membrane. Whether or not a substance can enter a cell depends on properties of the cell membrane as well as those of the substance. Cell membranes in the human body are selectively permeable, which means it allows some molecules to pass through while others are not allowed to pass through. Now, if a membrane allows a substance to pass through it, the membrane is said to be permeable to that substance. If it does not allow the substance to pass through, we said membrane is impermeable to that substance. Now, two properties of a molecule will dictate its movement across the cell membrane. First of all, the size, how big the particle is. And second, it's lipid solubility. Very small molecules like oxygen, carbon dioxide, as well as some lipid particles that are lipophilic, are able to easily cross most cell membranes. Okay. Whereas larger or lipophobic molecules, especially ions, which are charged, are not able to diffuse through that phospholipid bilayer, and so they require protein embedded within the membrane to cross, to carry them, or to let them to pass through the cell membrane. And very large lipophobic molecules that are too large for membrane proteins has to exit the cell via structures called the vesicle. Now, there are different ways of categorizing how molecules move across membranes. One way separates it according to the physical requirements, whether it moves by diffusion directly through the phospholipid bilayer. So that's our simple diffusion. Or does it require the aid of a membrane protein? That's our membrane-mediated transport. Or does it enter the cell in the vesicle. So that's our vesicular transport. And as we'll see, we can also categorize membrane transport by whether or not it requires energy. So that's our passive transport versus active transport. And in active transport, there are more subclasses that we'll go into. Now this tree diagram shows the categorization of membrane transport. You can see here that the categorization can be done based on whether or not the process requires energy. So if it does not require input of energy, we call that passive transport. And active transport will require energy. And as you can see, the vesicular transport of really large particles is always an active process. It always requires an input of energy. While simple diffusion of small particles or nonpolar particles like oxygen, CO2, that's always a passive process. And then we also have these protein-mediated transport, which can either be active or passive. Okay. And protein-mediated transport can either use a protein channel or it can use a carrier protein. We're first going to talk about diffusion. So some properties of diffusion. When molecules start out concentrated in one area of an enclosed system, their motion causes them to spread out gradually until they distribute evenly throughout the available space. And this process is diffusion. And so, in other words, diffusion can be defined as the movement of molecule from an area of higher concentration of the molecule to an area of lower concentration of the molecule. And this process is always passive, meaning that it does not require an input of energy from outside source. And that movement of molecule will continue until the concentration is equal on both sides. And as listed here, diffusion, the rate of diffusion can change depending on a few factors. Diffusion occurs faster when there's a steeper concentration gradient, meaning the concentration of solids across the membrane is drastically different. It's also faster across shorter distances, at higher temperatures, and also for smaller molecules. And diffusion can occur either within an open system, or it can also happen across a partition, such as across the cell membrane. And when we talk about the diffusion of ions, we also have to account for the distribution of electrical charge. And this is a concept we'll discuss in more depth in this later part of the lecture. So I want you to take a moment to watch this animation here. So first notice that in this image, okay, blue is potassium, which is positively charged. And green is chloride, okay, and these are ions. And notice that potassium is more concentrated on this side than on this side. That means we have a gradient that's going to want to draw potassium down its concentration gradient to this side, okay? However, as potassium begin to leave this side to go to this side, more and more positive charge will begin to accumulate over here. And what's left behind is a negative, relatively negative charge is going to be left behind as the potassium leaves, okay? And with that accumulation of negative charge, that negative charge exerts a pull on those positively charged potassium ion, okay? And so we say that the electrical gradient is going to help want to draw a potassium toward this side. Okay, the point in which that force, that chemical gradient drawing potassium toward this side, exactly balances the electrical gradient drawing the potassium to this side. That's what we call the electrochemical gradient. When we have reached an electrical chemical equilibrium is when those two forces are equal. Now let's take a look at a specific type of diffusion called simple diffusion. In simple diffusion, remember that there is no membrane protein, and these particles are just going to freely move across that phospholipid bilayer on its own. The rules for simple diffusion can be summarized by the Fick's law of diffusion, which states that the diffusion of an uncharged solid across the membrane is proportional to the concentration gradient of the solute, the membrane surface area, as well as to the membrane's permeability to the solute. And so the rate of diffusion through a membrane is going to be faster if we have a larger surface area to cross. Okay, so that larger surface area membrane. Also, faster diffusion rate if we have a thinner membrane, a more steep concentration gradient, or a higher membrane permeability to that particle. And membrane permeability to the molecule depends on the molecule's lipid solubility, its size, so the molecular size. It also depends on the lipid composition of the membrane. So naturally, lipophilic molecules are going to more easily move by simple diffusion across lipids. Now we're going to talk about protein-mediated transport. In our body, simple diffusion across the membrane is only limited to lipophilic molecules and small molecules. So as we mentioned, oxygen, CO2, and some lipophilic molecules or hormones. The majority of the molecules in our body are either lipophobic, meaning lipid-fearing, or electrically charged, and therefore cannot freely cross membrane by simple diffusion. Instead, the vast majority of solids that cross membranes in our body cross it with the help of membrane proteins. And this is a process we call mediated transport. If mediated transport is passive and moves the molecule down its concentration gradient, and if the net transport stops when concentrations are equal on both sides of the membrane, then we call this process facilitated diffusion. If protein-mediated transport requires energy from ATP or another outside source and moves that substance against its concentration gradient, so from a place of lower concentration to a place of higher concentration, then we call that process active transport. Before we go further into talking about protein-mediated transport, it's worth discussing the classification of membrane proteins. For a physiologist, the classification of membrane proteins by their function is more useful than classifying it by their structure. So if we're to classify it by structure, we could see there are integral proteins, also known as transmembrane proteins, that span through the whole thickness of the membrane. And then on the other hand, we also have peripheral proteins that do not span the entire thickness of the membrane. That's less useful than classifying by function. So if we're to classify based on function, we've got structural proteins, not to be confused with classification by structure. And we also have a class called the membrane enzymes. We also have membrane receptor proteins, also membrane transport proteins. And these groupings, as you'll soon see, they are not completely distinct. Some membrane proteins have more than one function, And so they can be both, for example, they can be both a channel protein as well as a membrane enzyme at the same time. So for the purpose of this lecture, you'll see that we'll be focusing mostly on the class of membrane transport proteins. And these are membrane spanning proteins that help move lipophobic molecules across membranes. Okay, so membrane transport proteins, they can be channel proteins or they can be carrier proteins. Now, first of all, we're going to talk about channel proteins. Channel proteins are named according to what they allow to pass. So most cells of our body have water channels, as we discussed earlier in this lecture, and these water channels are called aquaporin. And there are actually many different subtypes of aquaporin. In addition, there are hundreds of different types of ion channels that have been identified by research. Ion channels may be specific for one ion, like sodium only, or sometimes it may allow ions of similar size and charge to pass. So an example of that would be the acetylcholine ion channels that allow both the sodium as well as the potassium to pass through. And these channels can either be open channels, meaning they're constantly open and never close, or they can be gated, meaning that there is some kind of mechanism that can seal off that opening. And this gated channel, these gated channels can have different triggers. Some are chemically gated. For example, we can have a ligand binding to the channel, causing it to open. Okay, it can be voltage gated, meaning it responds, its opening and closing is in response to the change in membrane potential. Okay, they can also be mechanically gated, meaning some kind of mechanical trigger. Okay, can cause these channels to open or close. Moving on to talking about carrier proteins. The second type of transport protein is the carrier protein. Carrier proteins, they bind with specific substrates and carry them across the membrane by changing its conformation. Small organic molecules, such as glucose and amino acids, are too large to pass through channels, protein channels. And so they're going to cross the membrane using carrier proteins. Ions are much smaller, and ions can sometimes, but ions, in addition to using ion channels to cross the membrane, sometimes they may also move across the membrane using carrier proteins as well. And some carrier proteins move only one kind of molecule, and these are known as unipore carriers. If it can move two or more substrates in the same direction, we call these symport. Okay, if it moves substrates in opposite directions, in this case, we're moving sodium in this direction and potassium in the reverse direction. So this is called an antiport. Antiports are sometimes also known by the name of exchangers. Now, facility diffusion has similar properties as simple diffusion. Both are passive, so diffusion itself is a passive process. Okay? And transported molecules moving down their concentration gradient, that process requires no input of outside energy. And that net movement of those particles will stop at equilibrium, when the concentration inside equals the concentration outside. So by definition, facilitated diffusion carriers always transport molecules down their concentration gradient. Active transport is a process that moves molecules against their concentration gradient. That is from an area of lower concentration to an area of higher concentration. So while facilitated diffusion can use either a channel or a carrier protein, an active transporter is always a carrier protein or a vesicle. We'll discuss the vesicular transport in a few slides. And rather than creating an equilibrium state where the concentration of molecules equal throughout the system, active transport actually creates a state of disequilibrium by making the concentration difference across the membrane even more exaggerated or pronounced. And as you can imagine, moving molecules against their concentration gradient from a place of lower concentration to a place of higher concentration, that process requires an input of outside energy. And the energy for active transport comes either directly or indirectly from the high energy phosphate bond of ATP, which we discussed in the previous lecture. Active transport can be divided into two types. We've got our primary active transport, or also sometimes called direct active transport. The energy to push molecule against their concentration gradient comes directly from the high energy phosphate bond of ATP. In the second type of active transport called secondary active transport or indirect active transport. In this type of transport, the potential energy stored in the concentration gradient of one molecule is used to push another molecule against their concentration gradient. And all secondary active transport ultimately depend on primary active transport because the concentration gradient that drives the secondary transport are created using the energy from ATP. The sodium-potassium pump is probably the single most important transport protein in animal cells. And that is because it maintains the concentration gradient of sodium and potassium across the cell membrane. And as you'll see in later this course, it's very important for the system to maintain that sodium and potassium disequilibrium across the membrane. This transporter is arranged in the cell membrane so that it pumps free sodium out of the cell while pumping 2 potassium into the cell for every ATP consumed. Okay, and these, as you can see, contribute to the establishment of high sodium concentration outside the cell and high potassium concentration inside the cell. And sometimes this is also called sodium potassium ATP pump or sodium potassium ATPase. The sodium concentration gradient with sodium concentration higher in the extracellular fluid and lower inside the cell. This is the source of potential energy that the cell can harness for other functions. For example, nerve cells can use that sodium gradient to transmit electrical signals. Epithelial cells can use it to drive the uptake of nutrients, ions, water, etc. And as we'll discuss here, membrane transporters that use potential energy stored in a concentration gradient to move molecules are what we call secondary active transporters. So secondary active transport uses kinetic energy of one molecule moving down its concentration gradient to move other molecule against their concentration gradient. The co-transported molecules may go either in the same direction across the membrane, meaning it's a symport, or it may also travel in the opposite direction in the case of antiport or exchangers. And the most common secondary active transport systems are driven by that sodium concentration gradient. So here we can see, as we just discussed in the previous slide, the sodium gradient, the sodium concentration gradient established by the sodium potassium ATP pump, right? So we're able to maintain a low concentration of sodium inside the cell, okay? And we have higher concentration of sodium here relative to inside the cell. And this concentration gradient, okay, is going to drive, is a source of potential energy that drives sodium going down its concentration gradient into the cell. And this kinetic energy of sodium going down its concentration gradient allows glucose to be carried with it through this co-transporter, the sodium glucose co-transporter. Okay, so in this case, you can see that the transport of glucose, right, relies on the concentration gradient of sodium. And that concentration gradient of sodium ultimately relies on this primary active transporter. So this process of glucose transport is secondary active transport, but it relies ultimately on the energy coming from an active, from a primary active transporter. All carrier proteins, whether it's active or passive, are going to exhibit three properties that result from the binding of a substrate to a protein. And those are specificity, okay, binding of for a single type of molecule, competition, competition among molecules of the same shape, okay. It's also, they're also going to exhibit saturation, meaning the rate of transport is limited to a number of available carrier proteins. And so here we could see the concept of transport maximum. So in the example given here, we've got our GLUT transporters, which is a family of transporters that are able to bind to and transport 6 carbon sugars or hexose. And that includes glucose, mannose, galactose, fructose, etc. And these transporters have binding sites that are able to recognize and bind to hexoses, these glucose, mannose, etc. But they will not organize or bind to or transport other particles, other molecules, even disaccharides like maltose. So that's specificity. Now this graph shows glucose transport rate as a function of glucose concentration. And when only glucose is present, you see this is the transport rate. But now when we have a competitor molecule, galactose, you can see there's a reduction in the rate of glucose transport. Okay, and this is related to this third property, saturation. And so this graph, as you can see, shows the transport can reach a maximum rate when all the carrier binding sites are filled with substrates.